Good afternoon. I'm glad everybody made it back from lunch, and I hope that your discussions on the Quantum Leap program went well. We look forward to that RFA and to get some funding into this new concept. So it's my pleasure now to introduce Dr. Lorelei Mucci, who will introduce Elizabeth Platz. Lorelei is an epidemiologist and a biostatistician from the Harvard School of Public Health, a PCF Young Investigator, and somebody that really does a lot for the foundation, and for that we're thankful. She always answers our calls. So, Lorelei, come on up.
Well, I'd like to thank everybody for a really lively discussion over lunch about the Quantum Leap Award. And when you think about, for me, when I think about quantum leaps in prostate cancer epidemiology, the person who most comes to my mind is Elizabeth Platz. Elizabeth actually did her training at the Harvard School of Public Health and is now a professor of epidemiology and co-director of the Cancer Center, Johns Hopkins Cancer Center Cancer Prevention and Control Program. She's really led a lot of the first-in-field studies looking at the role of novel infectious agents in the etiology and progression of prostate cancer. She was really the first to look at medications that might lower the risk of advanced prostate cancer, such as statin use and digoxin. And right now, she's leading an incredibly interesting project looking at the role of telomeres in tumors in the adjacent stroma and how it might be used as a biomarker of prostate cancer progression. And I think what makes Elizabeth so emblematic of being a quantum leaper is her role in building incredible teams of individuals across multidisciplines as well as across different cancer sites to answer challenging questions. So I'm delighted to be introducing her to you today, and I'm really excited to hear her talk. So thanks, Elizabeth.
Great. Thanks, Lorelei. Actually, I'm going to give her a hug, because that was a lovely introduction. Thank you. Thanks. So great. So thank you for having me here today. So I want to share a little bit about my experience with team science. So at some point, I too was a young investigator. So that was now back in the late 1990s. And I guess questions come up. How do you get started? How do you get to form a team? How do these come about? So I'm going to share my experiences. So you heard Dr. Blair earlier today say he was giving a rather autobiographical talk. Well, I'm going to do the same thing. So here's the approach that we've tended to use in our multidisciplinary prostate cancer research team. So my basic science and clinical colleagues test their hypotheses in various settings. They could be in cell lines, so very basic mechanistic type studies. They have interesting discoveries along the way. And what we've done is we've been able to take some of those discoveries and to move them forward into population and translational studies, so epidemiologic studies, for example. We then have findings from those studies, and then we circle back through an iterative process to be able to continue to understand, explain, and discover about the problem of prostate cancer. Okay, so we've thought back on this many times. How is it that we get to work together? Why does it work? Why have we been able to move the field forward using this combined population, basic and clinical science team approach? And so I wanted to highlight some of my colleagues. So obviously, I don't work with Jim Watson, but this was at one of our annual retreats for prostate cancer. And so Alan Meeker's represented, and also Angelo DiMarzo. So since the day I came to Hopkins in 1999, we've been collaborating together, and it's been wonderfully successful. Okay, so I wanted to talk a little bit about what are the attributes of the team members that led to our having some successes in doing team science for prostate cancer. And so the first one is obviously we have shared interests. We all care very much about the problem of prostate cancer. We want to be able to solve it. The next characteristic is that we have complementary expertise. So we bring to the table a rich diversity of approaches and perspectives. Okay, this seems obvious, but sometimes you have to have the right people in the room. Okay, you have to figure out how to reach out to those people in the other disciplines and bring them together. Another important point is we're not replicative in our expertise. Therefore, we don't have competition. Okay, so we're all able to work together for the same purpose, to solve the problem of prostate cancer. So another very important feature of our team's personalities is that we respect one another very much, and we respect each other's disciplines. So my close colleague Angelo DiMarzo and I have in the past discussed the idea of respect. So he's a pathologist, I'm an epidemiologist. We together and individually over time had experienced other situations where there wasn't respect for our disciplines. Our disciplines were seen as helper disciplines rather than sort of the active, actual doer of the research. And so we were able to commiserate over that and bond over it. So what we try to do is to make sure that we foster this notion of respect for other disciplines in our trainees. And here's a photo of Angelo sitting and showing our epidemiology and cancer control students, our population-based science students in training, doing a tour of prostate cancer under the microscope so they have an understanding of what he does for a living. Okay, another very important characteristic of our team members is generosity and trust. Okay, so we have a tradition of open exchange and of helping one another. And I think the major reason why we operate in this way is because of this man, Don Coffey. So we all acknowledge that he's the one who has really created this environment where we want to be trustful of each other. Okay, so he makes sure that we all can share our findings. We don't worry that someone else is going to steal what we're up to. Right? He's created this supportive environment for open exchange. Okay, personalities. We just happen to have personalities that melded very well together. It just works out. We want to collaborate. So there's Alan Meeker. This photo was actually taken by Pat Walsh. He likes to come and get photos of us working together, and he takes the photos himself because he thinks he can capture our personalities much better. So you can see it's a happy photo. Okay, incentives to be a team member, and this is a really important one, intellectual stimulation. When you bring together people from a variety of disciplines, you get to hear about what they do, you get to see how they think, hear the logic of their field. It's very exciting, it's very intellectually stimulating. So here's a really, I think, an interesting example of how intellectual stimulation actually led to something relatively novel. Lorelei alluded to this. Being able to work together, bring basic science and epidemiology together through a two-step process to think about whether there are drugs for other indications that may have utility either in the prevention or treatment of prostate cancer. And so I'm highlighting two of our colleagues, so Vasan Subramanian and Bill Nelson, who were working already with June Liu, who's a pharmacologist at Johns Hopkins, to think about finding drugs for other indications that may be able to prevent or treat prostate cancer. And so they came to me one day very excited. They thought they had some hits, and they said, could we work together to be able to see if those same drugs could be associated with prostate cancer in an epidemiologic study? Okay, so again, this was an interesting idea. It's the notion of drug repositioning. And what we did is we used this two-step process. So the team screened through a library of FDA-approved drugs for those drugs that had cytotoxic potential against prostate cancer, both androgen-dependent and independent cell lines. And those that met a certain criterion for cytotoxicity, they then moved forward into another series of prostate cancer cell lines. And then what we did is we took the top hits, so those that were the most effective at the lowest concentration, and those that were in common use in the US population. So a couple emerged, a couple of classes. And so obviously we weren't interested in looking at the known chemotherapy agents, so we put those aside. So the ones we focused on were those that were not known to be chemotherapy agents. Okay, so if you look at the panel that's on your left, you can see that the second item down, second drug, is digoxin. And so we thought that was sort of interesting. So that's a drug, it's a cardiac glycoside. It's used to treat congestive heart failure and arrhythmia. Okay, there was another drug on that list that acted on the same enzymes, a sodium-potassium ATPase inhibitor. And there was another drug that actually didn't make the list because it wasn't FDA approved, but it also inhibited the same enzyme. So we thought that was kind of interesting. Turns out digoxin still has common enough use in the US population, at least in the cohort that we were going to study, to be able to make this a feasible epidemiologic study. So here are some results. So what we found when we moved into the epidemiologic study, which by the way was a large cohort study, of health professionals. So it's a Harvard cohort. That's where Lorelei mentioned that I trained. And it's great that they still consider me to be a co-investigator, which then allows us to do interactions across disciplines, including the work that Lorelei alluded to on telomere length and prostate cancer mortality. So in any case, you hear the results for digoxin. What we found was that men who used this drug had about a 25% lower risk of developing prostate cancer in the future. And then when we looked at the duration of use of digoxin, men who used the drug for a longer time had a lower risk of the disease. Okay, so this was an interesting result, we thought. And so this was a two-stage transdisciplinary approach. We went from one discipline right into another discipline. We did it together. We think this drug, or at least the pathways that you might think the drug affects, are interesting candidates for further mechanistic study. And perhaps at some point, there might be some type of clinical trial that could be done, maybe not using the drug itself, but targeting some of the pathways the drug influences. Okay, and so we thought that the significance of this work is beyond that. We think that by putting these two types of designs, two types of studies together, the basic science piece with the observational piece, works sort of like screening. So if you do screening in sequence, in series, the drug would have to test positive in the first series and then test positive in the second one as well. So it has to be positive twice, which means that we probably reduce the likelihood of a false positive hit. Okay. So we were thrilled that there was a lovely editorial written about this work called Epidemiology Found in Translation. And these editorialists made the point that they thought that this was a wonderful use of epidemiology, a very modern translational approach to using the epidemiologic thinking and methods to ask important questions about cancer. Okay, so moving back to our thoughts on being a team member. Okay, so what are the incentives to be a team member? So I talked about the attributes of the team members, but what are the incentives to participate? Well, an important one is that success begets success. So as we began to have success, meaning we were writing papers, we were writing grants, things were getting published, we were getting grant funding, it kept us going. So once it works, we figured out how to continue to make it work. And it turns out that mutual benefit is really a powerful incentive to keep going. I think if we had failed early on, we probably would have moved on to other things. But because we had success, we kept going, and we continue to keep going. Okay, another really important incentive is satisfaction in together training the next generation. So here's Alan Meeker and me with two of our trainees who aren't so youthful any longer. They're young people, but what I mean is they're not at the very beginning of their career. They're moving on, right? So Corey is an instructor about to move up to the next rank, and Chris Heaphy is about to move up to sort of that next level, early faculty as well. And so we together formally and informally train fellows and doctoral students. It helps energize our team, and it perpetuates the team, and it also helps perpetuate the multidisciplinary approach. Again, that fostering our sense of respect that our trainees have for these disciplines. Okay, so how about institutional factors? What does it take to really cause team science to happen, to make team science productive and successful? So there needs to be vision and strong leadership. Okay, and this has been true historically and at present at our institution. And even before I got to Johns Hopkins, I knew that it's a place known for prostate cancer research. And so, again, a nod to Don Coffey. He's always brought this senior leadership and vision to our environment, as well as the national and international environment for prostate cancer research and cancer research in general. Okay, other institutional factors, very important, endorsement. Okay, so our highly respected senior investigators, senior faculty, have endorsed the individual members of the team to one another. And that causes us to want to work together. If someone says to you, this person is a wonderful investigator, this person is doing great work, that makes you want to work with that person. So endorsement is incredibly potent in getting teams to assemble. But then there's a little caveat. The team that we developed arose organically. We weren't forced to work together. So sometimes There's the idea if you bring people together and you force them to work together, something good will come out of it. That approach I don't think always works. These organic approaches where people's personalities meld, I think maybe a more natural way for a team to come together and then to have success. Okay, and then I wanted to give a little bit of my own personal history. So how was it that an epidemiologist, a population scientist, became embraced by basic and clinical investigators? Because this was not historically the case in most institutions. So how did this happen? So I'll tell you the very beginning story. So I had started to do a little bit of thinking about infections and prostate cancer. So Ed Giovannucci is shown here. He's my mentor up at Harvard. I trained with him. And he was the one that sort of encouraged me to think about prostatitis, infections, inflammation, and the disease. And he at some point said, would you like to speak at Proutsneck, which is a meeting that used to be held. I guess Hopkins organized it with other groups. The NCI, I guess, helped support it. And so I was a young person, and he said, they're looking for young people to speak. Would you like to go to the meeting? And I said, okay. And then when I got to the meeting, Ed knew Bill Nelson quite well. And Bill, of course, knew and had encouraged Angelo to think about the problem of inflammation and prostate cancer. And so what Bill and Ed did is he made sure that Angelo and I got to meet each other. And this was before I showed up at Hopkins. I was about to arrive. And so there was already this natural endorsement that happened. So when I did arrive, I was eager to start collaborating with Angelo. And I hope he was eager to start collaborating with me. Okay. And then it worked. And then it turned out lots of people wanted to collaborate with a population scientist. Okay, other institutional factors. And this one is a tough one. This one still needs to evolve. And that is the recognition of scholarly efforts of individuals participating in multidisciplinary teams. Okay? So somebody has to be first, somebody has to be last. But in a multidisciplinary team, you might have several senior investigators and several junior investigators. And where are they all going to go on the authorship list? And how can you make it clear that people are contributing equally? Okay? So at our institution, this has been recognized. Okay? How about institutional and national infrastructure? Okay, so how do we get teams to come together? Are there spaces where people can meet each other and network? Well, at our institution, there's certainly important venues for exchange of ideas. Okay, so we have something called the Long Rifles sessions. And so again, this is a Don Coffey idea that he put on the table years ago, and he's continued to foster and encourage these sessions. So we had one a good few years ago where we thought about whether genetics or the environment was more important in the etiology of prostate cancer. And so Bill Isaacs took the idea of genetics being more important, and I took the idea that modifiable factors were more important. To have an extreme set of views. And we started asking questions. And during these exchanges, Don will say, Platz, what do you think of that? Meeker, what do you think of that, right? And people will throw out some ideas, and it's this open environment for information exchange. Well, one of the ideas that came up was, what do we tell men with prostate cancer about what they may do to reduce their risk of having a poor outcome? And we actually called him Mike Carducci, who's here somewhere over that way. And Mike said, well, I tell them about eating well and stopping smoking if they're a smoker, and make sure they lose weight if they're heavy. And then we said, well, how do you know? How do you know if that's the right set of guidelines? Because we hadn't looked in men who already had prostate cancer to see if these are the right messages to give them to help them think about reducing the risk of poor outcome. And so it was this environment where we could challenge one another. And because of that discussion, Corey, who at the time was a postdoc, said, well, can't we study that? And we said, well, that's a great idea. We should study it. And so she began embarking on research on modifiable risk factors for poor outcome in men with the disease, with the idea of intervening on those risk factors to reduce poor outcome. Okay, so we have these venues for exchange that are incredibly important. Okay, so here are Corey's findings. What she actually saw was that men who gained weight from before they had their diagnosis to the time after their diagnosis, and it was a fairly modest weight gain, was associated with about twice the risk of recurrence. And this is taking into account any differences in stage and grade at diagnosis. Okay, so it's an important potential public health message. And because of this work, Corey, when she applied for PCF funding as a young investigator, to explore the biology underlying these observations using a PathoEpi approach, which is a term I think Lorelei and Epidemiologic coined at some point. She was funded, and so we're very excited about that for her. Okay, so other institutional and national infrastructure issues. So research prioritization. So we've had success in our translational research because right now translational research has such a major emphasis in the national cancer research agenda, especially for prostate cancer. And in fact, some funders require translational projects and cross-discipline projects. So we've certainly benefited from those requirements. Okay. How about some complexities, though, related to team science? Because it's not perfection, right? It's hard to do team science. So what are some of those complexities? So the first one is it takes a lot of energy. It certainly takes a lot of money, opportunities for unforced exchange of ideas, demonstration of success and mutual benefit, among other factors. Okay, so the energy is the big part. Making sure everyone keeps their energy level up to continue the process. Okay, get that critical mass going. Another really key problem that comes up is there's so many cooks in the kitchen when you do multidisciplinary research. Somebody's going to make sure the research actually comes to fruition, it gets written up, it gets communicated. Okay? So you can really get jammed up with all kinds of great ideas, start working on it, and never really move things to the end because there's so many people in the kitchen. Okay? And so that's this point. So many great ideas generated, so little time to pursue them because of all the different perspectives coming together. We get this backlog of great ideas. Okay, now this one is really a problem that's come up, we've faced a number of times. Reviewers are not yet prepared, still not yet prepared, to be able to review studies that take a multidisciplinary, interdisciplinary approach. Okay? We've had comments on our grant applications that say, I get the basic science mechanistic part, but that other stuff, that observational people stuff, toss it out. That's merely correlative science. Okay? So, you know, this is where we were trying to do a translational type study. Okay? Another problem is that the financial systems are not yet prepared. So when you try to do research within your own institution, but say between a school of public health and a school of medicine, they have different cultures for handling the finances. Okay? So they don't even use the same Excel spreadsheets. They don't even have the same financial systems. So it actually is a problem. It's been a barrier that we've encountered over and over again. Okay? So teamwork is quite powerful, but we still need to make sure we're addressing the right questions. And when I say the right questions, I mean the important questions. When you work as a team, there's so much to be distracted by. There's so many things that you could be pursuing, but it may not all be important. So what you need to do is think big and think about making a difference. So the quantum leap notion that we're talking about here today, okay? Leaps may come, especially when you have a team setting, because of the richness of the multidisciplinary perspectives of the team. Okay, so quantum leap, we were trying to define it over lunch, something that team science I think is really critical as part of the definition of what a quantum leap award ought to be. Okay, addressing the right questions. So how many of you saw this, this brief piece in Science not too long ago? Okay, I think this was really quite fascinating. It was about helping our trainees find a good set of dissertation questions. And what we tend to do is we tend to encourage our young people to think about filling the gap or figuring out what's the next increment in a line of observation, a line of reasoning, a line of inquiry. And what this particular group thought we ought to do is not that. We need to have our young people start thinking big from the beginning, think about making a difference. And what they actually alluded to is something called type 3 error. Okay, and they define that as finding the right answer to the wrong question. Okay, so I think we need to take that to heart. Okay, and we do have this obligation to address questions, the right questions, the important questions together to actually make a difference for the clinical situation of prostate cancer as well as the public health problem. And so I just bring us back to Corey for a minute. I mentioned that example from the long rifle session. She did something about it, and then I can tell you this is now a national priority to understand the role of obesity, weight gain, physical activity, inactivity in the health of cancer patients and cancer survivors. In fact, there was even a National Academy of Sciences workshop in the fall on this topic. Okay? So not to say that we shouldn't be conducting plain old etiologic research, pure basic science. We still need to do that if it's foundational. But what we need to do for all disciplines is avoid me too science. Okay? Okay? Ask good questions, important questions, questions that really do have the potential to make a difference. And so I want to give you a couple of examples using translational cancer epidemiology. So we should be conducting research on prevention, and here I actually mean tertiary prevention, and also prognostication for prostate cancer patients and survivors. And so we already gave you the example of modifiable factors in prostate cancer outcomes. But think about including an epidemiologist, a population scientist, when you're doing studies on prognosis. Population scientists bring design, they bring populations, they can show you how to even design a study using your own populations. Okay, so use cancer epidemiology thinking and design to enhance that type of research. Okay. We need to start thinking about expanding which disciplines we include in our team. So far we've included the obvious disciplines, but we've left out important ones like engineering and physical science. So we need to start bringing those in. In fact, they came to me recently. So we have a Center for Cancer Nanotechnology, and they had a symposium, and they invited me to speak, and I couldn't understand why I was there at first. And then I realized because they really were reaching out. And then it became much more apparent how we should collaborate. So it's very important to bring them in. We have to learn their language. The other group that we've not really thought so much about interacting with are the people who actually do interventions. Okay, so people actually go into populations, whether they are patient populations or the general population, with important public health messages. Figure out how to actually make a difference. So you heard Mike Milken talk about the problem of obesity. So even basic scientists, we need to think about how do we go into populations and intervene on the problem of obesity, because it is a risk factor for aggressive prostate cancer and for fatal disease, as well as a whole wealth of other chronic diseases. Okay, so working together, this is in summary, to be able to address important translational prostate cancer questions. There's great value in it for the individual researchers, for the team of researchers, for science in general, and for patients and the population. These approaches do have their complexities, but these are not roadblocks to success. And then importantly, and please everybody laugh, it's actually really fun when it works. And by the way, that's me. So this is teamwork with my personal trainer learning how to do something called acro yoga. So I thought maybe you'd remember the talk if you had something silly at the end. Teamwork, it can really make a difference.
Be nice if we could see out there.
I know. This
Dr. Coffey has a question.
Light here.
Liz, I think you gave a nice view of what it's about, but I want you to share with the audience something that's just stunned me. What it is that if you take top flight people doing gene searches in prostate, and another institution does it, and you do the diagrams, they don't overlap the same genes.
Right.
And all of a sudden the antibodies, and done in a big company, from 4 top institutions with 1,000 in each one, and the blood run at the same time, none of the antibodies overlapped.
Right.
So you're going to have a long rifles on that. Tell us how you'd approach that question, what that means to this audience, because if that's true, and it is, I just come from another meeting where they've shown that these things that we're showing cannot be reproduced in a way between 2 labs.
Yeah. So first thing I need to say is it's frustrating because it's so much money goes into this type of work, and yet it's not clear what the answer is going to be. All right. So what Don's talking about is where you have multiple institutions with their own samples, and everybody takes a look, say, across the entire genome, okay, using an agnostic approach, okay? So we don't have hypotheses going in other than there might be some variant across that genome that's related to the disease that I care about. When you use such large-scale agnostic approaches, you're going to get lots of false positive hits, okay? And those false positive hits may or may not overlap across all the studies that are using it. That's right, with tiny p-values, but those tiny p-values can still be chance, right? They still can be chance, all right? It's because the sheer total number of looks that were taken across the genome. And so now what many groups are doing is they're going back and they're trying to follow up to try to understand, well, what's going on in that one particular place? Is it real? Is it not real? Okay, so one source one explanation for what you're pointing to is just chance. Okay, that's the first one. The second one is each group is using their own patient or participant populations, okay? And there could be differences among those populations that are real. So those could be real differences. But again, because the approach was agnostic, we don't know, we can't discern between what's real and what's due to chance. And so again, that involves going back and actually comparing the patient populations and trying to see if there are differences that could account for it. And so what Don's talking about, the long rifles, is we're going to have one on biomarkers, hitting the target rather than missing the target. And what we want to do is emphasize making sure that you know who you're studying, you understand what the patient population is, how it relates to the other patient populations and real patients at the current time, understanding study design, making sure we're using appropriate approaches, making sure that we understand even pathology. Imagine how important it is if people are looking for not germline differences, but tumor-based differences, somatic events. Well, making sure you have proper sampling of the tumor and not incorporating other cells that are nearby. So there's so many ways that these kinds of studies might have problems. It's not that I'm not going to call them flawed, but there are so many places where problems can creep in. We need to make sure we do these types of studies in a very standardized way if we're not going in with hypotheses up front. Thank you for listening.